We're going to be doing pre-cell energetics. There's a lot to get to. I hardly finished my first period, so let's go and get started. I'm recording the lesson right now. Here we go. The first thing we're going to talk about is enzymes. We talked about them on Tuesday, but we're going to do it again. Enzymes are proteins. This means that they are composed of carbon, hydrogen, nitrogen, oxygen, and sulfur, and B, they're composed of amino acids. Enzymes are biological catalysts. A biological catalyst does not, any catalyst for that matter, does not get used up in a reaction, but it helps reactions go faster and use less energy. That's the whole idea of the catalyst, all right? So the first question I want to know is, because it is made up of amino acids, what would happen if there were substitutions to amino acids? What would that do? Well, it's just like some of your names. Like if I took the name Elizabeth, and if I replaced a letter with another letter, is that going to change how I say the name? Elizabeth. Well, what if I replace the Z with an S? Is that going to change how I say Elizabeth? Not really. This would be known as a silent mutation. Well, in biology, you have the amino acid, let me look it up here. You have the amino acid, let's say leucine. Well, a substitution would be having UUA, which is the amino acid leucine, and have a mutation where you have UUG. Now, you all should know that there are 20 different amino acids, but when we were using your codon charts, which you all should know how to interpret, the codon chart has 64 different combinations. So there are a lot of amino acids that have multiple combinations for the same thing. So UUA codes for leucine. It just so happens that UUG also codes for leucine. So if the A becomes a G, that would be no change. That would be a silent mutation. Well, let's say another mutation is encountered, and you have AUG. So now the U became an A. That would become methionine. That would definitely be a mutation that would change the sequence of the amino acid. This is known as a missense mutation. That would be like taking the name Elizabeth and beginning with a T. That would definitely change how we say it, Elizabeth. So that's not the same as it once was. This is a missense mutation. Well, that's what would happen if there was a substitution that actually happened to an amino acid. It could have an effect, or then it could not. It really just depends. In Elizabeth's name, if the Z is substituted by an S, that really doesn't change it. But if the E is substituted by a T, that would really change it. So that is what a substitution mutation can happen. What would that lead to? That could lead to the enzyme not folding properly. And that could result in loss of function. Got it? Now, another way that an enzyme can actually lose function is being denatured. Denature is a change to a protein, which includes enzymes, due to unfavorable conditions. These conditions could be where it's too hot or too acidic. So this includes things like temperature or pH levels. All right, let's give you a moment to catch up. Well, I would imagine most of you know what denaturing is, but you have to also know what it leads to. Denaturing can lead to the unfolding of proteins. Remember, the folding is what gives them their function. And so that can lead to loss of function, where the enzyme can actually slow down or stop. All right. The next thing I'd like to go over with you are exchanges of energy when you're actually using reactions. This is going to be really important for you to know. This is more of a chemistry lesson right now. So I want you guys to draw 3 axes like this. So just draw 1, then 2, then 3. I'm going to stick with 1 because I can erase stuff. But draw 3 of these. We're going to have the progress of the reaction. Your x-axis will not change. And then we have your y-axis. All right, so the first graph, I want you to draw an energy curve like this. This is known as an exothermic reaction. Exothermic is energy being released. That's what X means. We get that word for, we get our words exit or exoskeleton from the Latin prefix X. These are your reactants. These are your products. The energy, the very top, this little peak, is the actual chemical reaction. This is called your activated complex. And from the activated complex to that dotted line I drew is the activation energy. It's the energy needed to actually achieve a reaction. Remember to listen to what I'm saying. Don't just focus on what I'm writing. You know, I'm saying a lot of important stuff too. So I'll give you time to catch up on what I'm saying, but don't just focus on what I'm writing down. Also listen to what I have to say. So hear me too. The energy that you need to actually achieve a reaction is called the activation energy. And then the difference in energy from the reactants to the products is called your change in enthalpy. I'm actually teaching this to my chemistry students right now. If you hear the weed whacker outside, just ignore it. We're going to move on. Show goes on. Because there's an energy loss, that value for the change in enthalpy would be a negative value. All right, so I'll give you a moment to get that finished, and then I'm going to erase this graph, and I want you to move over to your second graph, okay? So I'll give you just a few seconds to get that finished up. All right, let's draw the second graph. Remember, I have the ability to erase all this. You guys really don't. Next graph is going to look like so. We've got reactants, we've got products. Notice that there's an energy increase. You end with more energy than what you left off with, or than what you started with. So this is known as an endothermic reaction. Okay. From the reactant energy level to the activated complex is your activation energy. Once again, this is your activated complex. And the energy from your reactant energy level to your product energy level is the change in enthalpy. And because it's going up, this would be a positive value. A mistake I've seen a lot of students make is that they try to say the activated complex goes from the product energy level to the activated complex. I'm going to show you what the wrong answer is. Don't do this. This is wrong, 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 wrong. I've seen this so many times. That is not correct. You are saying that the amount of energy of the products is what helps make the products. That makes no sense. You need energy from the reactants to make the products. Okay? So there's my second one. I'll give you a moment to get that drawn. And then let's get to our third graph. I asked you to draw 3 XY axes. Here's your third one. I'm going to erase my work. All right. Oop, sorry, excuse me, wrong one. Exothermic. This is going to be a graph without a catalyst. And then here is one. Oop, went a little too far down there. This is with a catalyst or an enzyme. I want you to notice 2 things. There's less energy used, less activation energy, and less time. The x-axis is time. That's the whole point of a catalyst. If we were in a regular school year and all of you came in person, a little demonstration I would have done with all of you is I would have fed you all one Ritz cracker. And I would ask you to put the Ritz cracker in your mouth and let it just sit on your tongue. Don't chew it. Don't crush it with your tongue. Just let it sit there. And wash your saliva all over it. This is an example of a catalyzed reaction. Ritz crackers are made of amylose, which is starch. It's a polysaccharide. Well, your saliva has amylase. You may have heard it called salivary amylase. Amylase breaks down amylose. And so in the matter of just a few minutes, that cracker would have been broken down into maltose, which is a disaccharide. So you got a big old polysaccharide broken down to a monosaccharide due to the enzyme salivary amylase. This happens in a matter of minutes, and that cracker would have just gotten really goopy and gross tasting. You're like, oh, just, let me swallow my cracker already. That's going to be an example of a catalyzed reaction. If you had no saliva, that cracker would have sat there for days, just dry and just gross. But because of your enzymes, a process that would take days happens in a few minutes. It uses less energy and it requires less time. Okay, let's get to enzymes again. Some things you guys should know. I'll put E for enzyme. This is your active site. And this is your allosteric site. Let's say that I have a substrate right here. Obviously, a substrate fits in there. Well, let's say that we have our enzyme. We have our substrate. We have an enzyme substrate complex. And the result of an enzyme substrate complex is going to be product. So I'll put P, product, and that's going to go do whatever it's supposed to do. The enzyme does not get used up. Remember that. The enzyme does not get used up. Well, what if you're making way too much product? The product is just overwhelming. You need to shut it down. You want to use something called an allosteric inhibitor. An allosteric inhibitor can fit into that active site like this. And it can actually change the active site so the substrate can no longer fit. So you see the substrate's like, oh, let me in. I can't get in. Enzyme says, nope, we're done. Well, what if we had the opposite? Let's say that we have an enzyme that is currently turned off. And we have a substrate that needs to fit. Right now, it won't fit. You would have to have something called an allosteric activator. An allosteric activator can fit into that allosteric site like so. See, it's fit in there now. And that's going to cause the active site to change. And the substrate can actually fit in there. So this is known as the induced fit model, where you change the active site so that a substrate can fit. Now, what if you're making way too much product? You just simply remove that allosteric inhibitor, or excuse me, allosteric activator out of there. And that would change the actual enzyme substrate or active site back to being incompatible for the substrate. But if you want to turn it back on, you just put that activator back in there, and that's going to change the active site. Okay? All right, one more thing. Let's talk about competitive and non-competitive inhibition. I'm sure you all have played musical chairs before. If you haven't, you had a horrible childhood. Just kidding. Competitive inhibition looks like this. Here's an enzyme. Here's a substrate. But here's an inhibitor. You can clearly see the substrate and the inhibitor cannot fit at the same place at the same time. So imagine that the enzyme is the last chair, and the substrate and the inhibitor are the last competing kids for musical chairs. There can only be one. And so they're fighting for that same spot. This is known as competitive inhibition. But if you had a enzyme that looked like this, you can see that the substrate can fit in here and the inhibitor can fit in there. This is known as non-competitive. Inhibition. Okay, so to finish off cell energy, remember that they're proteins. They can be denatured, which can result in a loss of function. They are made up of amino acids. And if you have the wrong amino acid sequence, that can result in the active site changing shape. That can result in the enzyme changing shape and not being compatible for a substrate. One last thing I want to go over, I promise this is it for energy, and that's coupled reactions. Coupled reactions are just basically where A yields B, or A plus B yields C, plus D yields E, plus F, and so on. So let's say I have a reaction here, and then another one, and then another, and then another, and then one more, and then one more. If I ask you how many reactions do I have, well, let's count them up. 1, 2, 3, 4, 5, 6. Here we have an exo, this is endo, this is exo, this is endo, and this is exo. Okay, these are coupled reactions. So imagine that A results in B, B results in C, C results in D, D results in E, E results in F, F results in G. So a lot of you are seniors, and let's say that after your senior year, you have all this new knowledge, you have all this stuff that you know. This is going to help you get into college. And you're like, okay, all my work is paid off. I'm done. I've got all this stuff. I've accomplished my goal. Well, when you go to college, all that knowledge that you needed at the end of your senior year is now going to be used at the beginning of your freshman year of college. So what was once a reactant is now a product. Everything that you accomplished at the end of your senior year is now going to be everything that you need for the beginning of your freshman year of college. So what I'm saying is you have a reactant that led to a product, but when you move on over to the next reaction, this product is now going to be your new reactant. And that's going to lead to a new product. And the whole process just repeats itself. All right, let's move on to photosynthesis. Photosynthesis is all about capturing sunlight. To synthesize chemical energy. Remember, if I'm going too fast on this video, this is a video. You can pause it, you can rewind it, you can do whatever you need. If I'm going too fast, which I entirely understand, I probably am, but you have the ability to pause and play and everything you need to do, all right? The chemical equation is 6 molecules of carbon dioxide reacts with 6 molecules of water to produce, and sunlight to produce 1 molecule of glucose and 6 molecules of O2. Some of you would speak way too fast when I would ask you, what gas do plants need? So many kids would go, oxygen. No, they need CO2 for photosynthesis. They produce oxygen in photosynthesis, right? It occurs in the chloroplast in eukaryotes. Remember that the chloroplast is a result of the endosymbiotic theory. It used to be its own organism, which was a photosynthetic prokaryote. It has 2 stages. The light reactions, Rx and S is an abbreviation for reactions. This takes place in the thylakoid membrane. And the Calvin cycle, which takes place in the stroma. All right. I want to also have you guys understand, this one's really important. In the light reactions, you only need sunlight and water. You do not use carbon dioxide in light reactions. I've seen so many multiple choice problems on AP exams where they try to trick you into thinking that the light reactions need CO2. They do not need CO2. The Calvin cycle needs CO2. All right. Let's do a quick drawing of a chloroplast before I describe the 2 parts of photosynthesis so you know where we're talking about. This is a chloroplast. Here's the outer membrane. There's the inner membrane. This area, this open area is called the stroma. One of these little discs is called a thylakoid. The inside, carefully look where I drew my line, the inside of a thylakoid is known as the lumen, and then a stack of thylakoids is called a granum or grana. All right, let's get all that written out. Let's talk about pigments. You have chlorophyll A and chlorophyll B. These 2 are green pigments. Remember that a pigment absorbs all the colors that it does not reflect. Chlorophyll reflects green while it absorbs the other colors. So you should know that on the color spectrum we have Roy, G, Biv. Got some red, orange, yellow, green, blue, indigo, and violet. Plants are green because they don't absorb green. They reflect it. They also absorb some yellows, but mostly they absorb red, orange, yellow, blue, indigo, and violet. They reflect it. They reflect green, that is. That's why they appear green to us. So if you actually looked at a color spectrum, violet is the strongest color. Think of on the electromagnetic spectrum, the form of radiation that follows violet is ultraviolet. We all know that ultraviolet is the sun. It gives you cancer and sunburn. So violet is the strongest color. Red is the weakest color. If you actually looked at the color absorption, it looked like this. You see that green is not absorbed. because the y-axis is the absorption rate. The higher it goes on the y-axis, the more of that color is absorbed. All right. If you guys have any questions, don't hesitate to send me a chat. I can answer it. I'll try to do the best I can. Let's keep going. Let's do light reactions. Okay, here we go. We have photosystem 2. We have the electron transport chain, which is just like a series of little proteins along the chain. We have photosystem 1. And then we have the ATP synthase. All right, remember that this is the thylakoid membrane. So inside the thylakoid is the lumen, outside the thylakoid is the stroma. All right, well, we have the sun. The sun is shining down on this plant. This little yellow blob is going to be the sunlight. And so plants need sun for the light reaction, clearly. Light reactions, they need sun. The other reactant they require is water. And we're going to do 2 water molecules at a time. And when these water molecules go to photosystem 2, they break down into 3 things. One thing they break down to is oxygen. Oxygen is out of here. It goes to the atmosphere. It leaves the plant. We need it. Next is going to be 4 hydrogen ions at a time. So inside the lumen, you are generating a tremendous hydrogen ion concentration. We'll get to this momentarily. But as more and more water, because plants don't just get 2 molecules of water, you water a plant, you give it an uncountable number of water molecules. You got a hydrogen ion gradient. And then the next and final results from water are 4 electrons. And this is what I want to focus on first. These electrons get powered up by the sun in photosystem 2. The sound effect I would use during class would be the electrons getting powered up by the sun. It gets powered up, and then it travels down the electron transport chain. And when I say travels down, I mean it literally, it's like losing power. It's going down. And it makes it all the way to photosystem 1. The sun is then going to shine down on photosystem 1, and the electrons gets powered up again. And it will actually combine 2 electrons at a time and a hydrogen ion with NADP plus to become NADPH. NADPH is an electron carrier. Just like the mailman is a mail carrier and the pizza boy is a pizza carrier. This electron carrier is going to go to the Calvin cycle. Remember that I told you when we did this lesson, I want you to think of that P as plants or photosynthesis. NADPH is what is in photosynthesis. So there's what the electrons do, all right? Well, let's talk about these hydrogen ions. You guys should know that on the most basic laws of diffusion, you have a high concentration down here in the bottom. You have hardly any concentration on the top. You want to go from high to low. But we have a problem. There's a membrane right here. Look at this. This membrane is nonpolar. Hydrogen ions have charges. They're polar. They can't just pass through the membrane. They need a pump. That's what the ATP synthase is for. These hydrogen ions will pass through the pump, and every time they do, they produce ATP, which is energy. Now, by the way, ATP has the elements carbon, hydrogen, nitrogen, oxygen, phosphorus. So ask yourself, what macromolecule is it? Is it a carb, a lipid, a protein, or a nucleic acid? Your answer should be nucleic acid, and the dead giveaway is phosphorus. It is a nucleic acid. Well, this ATP will also go to the Calvin cycle. Got it? All right. So the last thing with the light reactions is that the production of ATP is known as chemiosmosis. So I want to be sure you guys understand the difference between a noun and a verb. The noun is ATP synthase. This is a place. You all should know that nouns are persons, places, or things or ideas. ATP synthase is a thing. It is an enzyme that makes ATP. Chemiosmosis is a verb. It is the actual process of making ATP. Riddle me this. If the ATP synthase complex had a substitution mutation that altered its amino acid sequence, would that affect its ability to produce ATP? Yeah, it's an enzyme, which means it is a protein, which means it is made up of amino acids. A protein is made up of a very specific sequence of amino acids. If you alter that sequence, it's not going to work properly, or it's likely not going to work properly. All right, let's catch our breath here. There's still a lot to go, but that's generally the light reaction. I want to remind you that on my YouTube channel, I have every video since September post on there. So if you want a more in-depth look on light reactions, it's under Unit 3 on that playlist, okay? But now let's get into the Calvin cycle. The Calvin cycle takes place in the stroma, and it follows the light reactions. So all that ATP and NADPH that was produced in the light reactions is used in the Calvin cycle. Well, in the Calvin cycle, you basically have three parts, and I'm not going to go too in depth on this, everybody, but the three parts are going to be CO2 fixation, carbon reduction, and regeneration of RUBP, okay? Those are the three parts of the Calvin cycle, but here's basically what's happening. This may ring a bell from last October. We have 1, 2, 3, 4, 5. 5 carbons. This is RUBP, a 5-carbon molecule. If you really want to know the name of RUBP, it stands for ribulose bisphosphate. Ribulose bisphosphate will combine with CO2. You should know that CO2 is 1 carbon molecule. So 1 plus 5 results in 1, 2, 3, 4, 5, 6. A 6-carbon molecule. This process happens because of the enzyme Rubisco. Rubisco is an enzyme. It is what fixes carbon from CO2 onto RUBP. When I say fix, I mean added to. This Rubisco actually stands for ribulose bisphosphate carboxylase. I think you would rather use Rubisco instead. Well, Rubisco will join RUBP with CO2, and through a whole bunch of processes that you don't really need to worry about too much, you're going to use NADPH. Remember that when it gets oxidized, it returns to being NADP+. This will then go back to the light reactions to get recharged. So it goes to the light reactions. And ATP will join in here. The ATP that you made from the light reactions. It's going to get reverted back to ADP plus a phosphate. And then that will go back to the light reactions to get recharged as well. And it's going to reload. And essentially, eventually, you will make 1, 2, 3, 4, 5, 6. You'll make glucose. That's the whole goal here, right? This takes 2. You have to go through the 2 times through the Calvin cycle to make glucose. You have to go through the Calvin cycle twice in order to make 1 molecule of glucose. And this is what gives the plant the sugar it needs for starch, for cellulose, for all the energy it requires. Okay, we're almost done with photosynthesis. We have about 16 minutes. Now, there is an issue with Rubisco. I want to draw down here so I'm not so cramped. 1, 2, 3, 4, 5, 1. As you all know, again, there's RUBP, there's CO2. They come together to make a 6-carbon molecule. 1, 2, 3, 4, 5, 6. That is not glucose, by the way, not yet. This is done by Rubisco. Well, here's an issue. When temps are high, evaporation rates are high. Plants lose water, which is not good. This is known as transpiration. When plants have water evaporated from their stomata. So what plants do is that they close the stomata. The stomata are the openings on the bottom of a leaf. Now, this results in something good. No more water loss. You know, that's great. Happy. But here's the downside. O2 can't leave. And CO2 can't enter. That's bad. So that's, you know, double bad and double sad. So here's what happens. Everybody look over here. CO2 is no longer in the cell because the stomata have been closed. You're going to use oxygen instead. This is known as photorespiration. This is like a plant's version of fermentation. This is where Rubisco fixes oxygen to RUBP instead of CO2. The reason why is because CO2 is unavailable. This is highly inefficient and it's actually very dangerous for the cell. So there's a more evolved form of photosynthesis called C4 photosynthesis. C4 photosynthesis means a 4-carbon molecule. So in C4 photosynthesis, you have a 1, 2, 3 carbon molecule called PEP. And then you have a 1 carbon molecule called CO2. The two then come together to form a 4-carbon molecule. That's why it's called C4. This is called oxaloacetate. This is accomplished by the enzyme pepcase. And here's the great thing about pepcase. It doesn't touch O2. So it avoids photorespiration. Okay? That's called C4 photosynthesis. Pepcase binds PEP to CO2 to make a 4-carbon molecule, and then that will eventually lead to RUBP. So you don't have to worry about photorespiration. Then lastly, we have CAM photosynthesis, which is basically during the day, you have your light reactions. Stomata are closed. Okay? Because the evaporation rates are so high, stomata are closed. But at nighttime, you do the Calvin cycle, and the stomata open. This will allow CO2 in and out, and you must remember, the Calvin cycle uses CO2. So when the light reactions are going on during the day, the fact that the stomata are closed is not a big deal because CO2 doesn't need to come in yet. You're not doing the part of photosynthesis that needs CO2. The Calvin cycle needs CO2, but the Calvin cycle isn't happening during the day. So the absence of CO2 won't be a big deal. Once the sun sets, the stomata open, the Calvin cycle will proceed, and CO2 will be pumped in and O2 will be pumped out. All right, that is it for photosynthesis. We have 10 minutes to do cell respiration. So we're really against the gun here. Let's try to get as much done as we can. This is the production of ATP for cells. It's in the mitochondria. At least the aerobic part is. And this is only in eukaryotes. All eukaryotic cells have mitochondria, plants, fungi, animal, and protists. The equation is 6 molecules of oxygen react with 1 molecule of glucose to produce 6 molecules of H2O and 6 molecules of CO2 and a lot of ATP, like 38 ATP. Okay, this takes place in the mitochondria. Let's do a quick drawing on the mitochondria. Most of it takes place in the mitochondria. We have the matrix inside. We have the inner membrane. This is also called the cristae. We have the intermembrane space. And then we have the outer membrane. All right. Cell respiration begins with glycolysis. Glycolysis is in the cytoplasm. And the fact that it is in the cytoplasm means that it is anaerobic. This is the production of pyruvate. To visualize this for you all, here's a glucose molecule. 1, 2, 3, 4, 5, 6. That's glucose. Glucose gets broken down into 2 molecules of pyruvate. It's important that we follow what that pyruvate does. All right, so glycolysis is in the cytoplasm. If you do have oxygen, so I'm going to say O2, check. That means it is aerobic. You'll go to cell respiration, which is in the mitochondria. But we're going to do something else first. Let's say that there is no O2. So O2 is not here. This would be anaerobic. You're going to do fermentation. Fermentation is much less efficient. It is in the cytoplasm. And depending on what type of organism you are, you could either do lactic acid fermentation, which is also called lactate, or you could do alcohol fermentation, where you produce alcohol. Now, glycolysis produces NADH. That is another electron carrier. The NADH will go to fermentation and get oxidized back to NAD+, and then return to glycolysis. So keep that in mind. NADH is made during glycolysis, but then it is reverted back to NAD+ during fermentation. Fermentation is breaking down pyruvate. Pyruvate is metabolized. If it's in lactic acid, it's made into lactate. If it's in alcohol fermentation, it's made into alcohol. But that's what happens if there is fermentation. But now let's say that there is actually oxygen. You're going to go into the mitochondria. The mitochondria has all aerobic processes. We have about 6 minutes. You have the prep reaction. You have the Krebs cycle, also called the citric acid cycle. Has 2 names. And then you have the electron transport chain, which is on the cristae of the mitochondria. I should mention that these 2 are in the matrix. I'll try to draw this fast. I know I'm running low on time. Basically, what you're doing is you're breaking down pyruvate more and more and more and more and more. And what they're doing is in the production of pyruvate, you're sending NADH to the electron transport chain, and you're sending NADH and FADH2. So keep this in mind, everybody. And I'll try to draw my NADH. I know I'm really stretching it. This is looking really complicated. I apologize, but that's just how this goes. NADH is made by glycolysis, it is made by the prep reaction, and it is made by the Krebs cycle, all to go to the electron transport chain. FADH2 is made only in the Krebs cycle. All right, and all this NADH and FADH2 are electron carriers. That's what these are. They're carrying electrons to the electron transport chain to result in ATP, about 38 of them total. Now, last thing I'm going to try to do is draw this electron transport chain for you, and that will have to be it, okay? So here is the cristae, which is the inner membrane. You have all these proteins embedded in, and then you have the big guy, the ATP synthase. This is the matrix, and this is the intermembrane space. Well, electrons are going to be traveling down this chain, and eventually they will be accepted by oxygen, all right? Oxygen is the final electron acceptor. I've seen that in so many tests. It will accept those electrons at the end of the electron transport chain. You also have a lot of hydrogen ions. Don't mind the leaf blowers. We're still getting the job done. These hydrogen ions are going to be passing through the ATP synthase to produce ATP. And now you have hydrogen over here. The oxygen will combine with the hydrogen to form water, which, as you know, is a product of cell respiration. All right, guys, that is about it. I know I threw a lot at you. There is a lot here, no doubt about it. Let me check to see if I hit everything I needed to go over. Yeah, I think I got it all. So if I were you, I would definitely go back, play, pause, play, pause. I know there's a lot to take in. But with that being said, that is all of Unit 3. Basically, that's a month's worth of material broken down into 47 minutes.